All right, section 3.5, the cytoskeleton, cell movement, and cell junctions. You should be able to explain the role of the cytoskeleton in the cell, summarize the major protein fibers in the cytoskeleton, describe the role of the flagella and cilia in humans, and compare the functions of adhesive junctions, gap junctions, and tight junctions in human cells. So, the cytoskeleton. The next thing that we're going to discuss today is the cytoskeleton. This is going to be responsible for maintaining the cell shape, assisting in the movement of cell and organelles, help make internal transport possible, and this is going to be done through 3 different types of macromolecular fibers: the actin filaments, intermediate filaments, and microtubules. These are going to assemble and disassemble as needed. So it's kind of like the bones of and muscle of an animal, but it's actually much more dynamic. It responds to environmental changes. So you can kind of think of it like, imagine you're sitting there and you're just a great big circular blob, and then you see the sandwich in front of you and you're like, oh, I want that sandwich. Let's make an arm real quick. And so your skeleton just like assembled an arm out of nothing. And then you use that arm, you brought that sandwich to your face, and then you put it in your mouth, and then once you were done, your sandwich is in your mouth, you're going to disassemble your arm and go back to being a perfect sphere. That's kind of how the cytoskeleton works. It assembles and disassembles as needed. So it's very dynamic, very fluid, which is just kind of really awesome. So I do want to talk just briefly about microtubules. Microtubules are hollow cylinders that are made out of 2 globular proteins called alpha and beta tubulin. And there are going to be spontaneous pairings of alpha and beta tubulin molecules form structures called dimers. Those dimers will then arrange themselves into tubular spirals of 13 dimers around. And all of this assembly, all of this control is done under the microtubule organizing center. The most important part of the microtubule organization center is the centrosome. The centrosome is going to play a huge role in DNA replication and the processes of mitosis and meiosis, which is why I bring it up now, just because we're going to see the centrosome again later. So the microtubules are going to interact with some motor molecules like kinesin and dynein, and they're going to cause the movement of organelles. This is really, really awesome. So this kind of acts as like the superhighway across the cell. And what happens is we have these little motor molecules like kinesin and dynein that are going to bind to the microtubules and actually walk along these microtubules while they're carrying their vesicle transport. This would be a really awesome video. So extra credit points to anybody who wants to send me a video of vesicles walking along microtubules using those kinesin and dynein receptors. So this is a really great opportunity for you to send me some extra credit videos. All right, so to continue on with the cytoskeleton, we also mentioned actin filaments. Actin filaments, you may also know them as microfilaments. They're very long, extremely flexible, and very thin fibers that occur in either bundles or mesh-like networks. They're very, very thin filaments, and there's going to be a dense web of them just under the membrane that's going to help maintain cell shape. And the interesting about this, you remember when we talked about the cell membrane and how some proteins can actually be anchored to the cytoskeleton beneath? They'll be anchored to those actin filaments. They're also going to be responsible for intracellular traffic control, for moving stuff around within the cell. They're responsible for cytoplasmic streaming in plant cells, which plays an important part of photosynthesis. They function in the pseudopods of amoeboid cells. So if you've ever seen an amoeba and how it kind of stretches out these pseudopodia to help it walk and creep along, this would be another awesome video for you to send me. Send me a video of a white blood cell creeping along or engulfing and ingesting another smaller bacterium. So they play a very important, they also play a very important role in muscular contraction. This is another place where we're going to see myosin functioning as a motor molecule. So anyway, those are actin filaments. They're also going to play an important role in animal cell division, but I will get to that a little bit later in this semester. So the intermediate filaments are next. The intermediate filaments are going to be intermediate in size, halfway in between those actin filaments and those microtubules. They're going to be rope-like. They're going to be responsible for assembling fibrous polypeptides, and they kind of vary in nature from tissue to tissue depending on what is needed. Their function is going to be primarily to support the nuclear envelope and for cell-to-cell junctions, like those holding your skin cells tightly together. Protein keratin also provides mechanical strength to the skin cells. All right, so we also have cilia and flagella. Cilia are hair-like projections from the cell surface that kind of help in the movement. They're very different from prokaryotic flagella. They're an outer covering of plasma membrane, and inside of this would be like a thin cylinder of 18 microtubules arranged in pairs of 9, and I'll show you on the next slide. In the center of this is going to be 2 single microtubules, and this 9-2 pattern is used by all cilia and flagella. In eukaryotes, cilia are going to be much shorter than flagella. Cilia kind of move in a coordinated waves, kind of like a row of oars on a ship. They're all going to sort of move together, and we're going to see them a lot in things like the cells lining your upper respiratory tract. They help move fluid past the cells in order to clean dirt and debris out of the mucus that's lining your upper respiratory tract. Flagella, on the other hand, are going to be used primarily for the propulsion of the cell itself. So you're going to see these a lot in like sperm cells. They move like a propeller or a corkscrew, and the idea is to propel the cell itself. So here's that 9-2 structure that I was talking about, where they occur in these 9 pairs of 2 wrapped around 2 central microtubules. And this is just sort of the motor of the cilia and the flagella. Outside of the cell, we have what is known as the extracellular matrix. And I do want to mention this because it does play such an important role in binding to the actin filaments of the cytoskeleton. So the extracellular matrix is a protective mesh of proteins and polysaccharides that's sort of like the insulation between each cell. It's going to contain things like collagen, which resists stretching, and elastin, which provides resilience. There's also going to be fibronectin. Fibronectin is an adhesive protein that binds to integrin. Integrin is an integral membrane protein that's connected to the cytoskeleton. This is what anchors all of your cells to each other and to the neighboring cells and to the extracellular matrix. So we have this integrin protein that connects the actin filaments, the cytoskeleton, to the extracellular matrix. It's also going to play a very important role in cell-to-cell signaling. So here's the summary for all of that. If you want to write that down, there you go. I just said all of this, so I'll let you read this and write this down on your own. All right, so the last thing I want to mention is the junctions between cells, especially because in this class we're going to spend a lot of time talking about various tissues and organs, and so it's important to know how those tissues are held together. So certain tissues of vertebrate animals are known to have junctions between their cells that allow them to behave in a coordinated manner, and this is going to be done with 3 different types of junctions. There's adhesion junctions, which mechanically attach adjacent cells with intercellular filaments between the cells. You can kind of think of this adhesion junction as a buttons on a coat. So if you have a winter coat, it'll be held together by these adhesion junctions, which are going to button up or attach one cell to another. One type of adhesion junction that you're seeing here is a desmosome. This is where we have an internal cytoplasmic plaque that's going to be attached to the cytoskeleton of each cell, and they're going to be joined by integral membrane proteins called cadherins. This is a really sturdy but also flexible. So you're going to see this for things like your skin. So the sheets of cells like your skin, they're going to be held together by sort of this button feature. We still have a little bit of space between cells. We still have some flexibility, but we also have some nice tight holding together. The other type of adhesion junction is the tight junction. This one right here, this is going to bring cells even closer together than desmosomes. So tight junction proteins actually connect the plasma membranes between adjacent cells together. So this is kind of more like a zipper, where we have these really, really tight junctions between the cells. You're going to see this in tissues that serve as barriers. So things like the intestines. In order to keep those digestive juices from leaking into the rest of the body, we want these really, really tight zipper-like Ziploc closures. So that'll be this right here. Now, another type of junction that's not really an adhesive junction. The point is not necessarily to glue two cells together, but gap junctions. Gap junctions are really awesome. They're designed for cellular communication. This is where we have, it forms when two identical plasma membrane channels join together. So we actually have a hole. We can actually have cytoplasm flowing back and forth from one cell to another. The channel of each cell is going to be lined by 6 plasma membrane proteins. And then gap junctions provide strength to the cells, so they do kind of button them together, but it also allows small molecules and ions to pass through them. This is going to be super, super important in like the heart muscle and smooth muscle, where it's important for ions to flow from one cell to the next to facilitate simultaneous contraction. When we want the whole heart to beat as one, we need rapid cell-to-cell communication. And that's going to be done with these gap junctions. So you should be able to list the 3 types of fibers in the cytoskeleton, describe the structure of cilia and flagella and the function of each, and then also list the types of junctions found between animal cells and state the function for each.